Good morning, dear students. Welcome to UDAAN Career Institute. In today's class, we will continue with the chapter that is mineral nutrition. As all of you remember that in the last class, we have gone with nitrogen cycle and various different processes that takes place in the nitrogen cycle. In today's class, we will continue with biological nitrogen fixation. Now, as students, as we have gone with nitrogen cycle in the last class, I have told you about the nitrogen fixation step. Now, nitrogen fixation was the first step of the nitrogen cycle, in which the nitrogen which was present in the atmosphere, it is brought into the soil, or it is allowed to enter into the soil. That process was basically known as nitrogen fixation. So nitrogen fixation was taking place either by nature, or it was taking place because of the industrial combustion or because of the firewood. Now, there is another mode of nitrogen fixation which also takes place, which is known as biological nitrogen fixation. Now, what is this biological nitrogen fixation? Now, there are certain prokaryotic organisms or species which are capable of fixing atmospheric nitrogen into the soil, means they can fix the atmospheric nitrogen into the soil. Now, reduction of nitrogen to ammonia by living organisms is known as biological nitrogen fixation. So, in biological nitrogen fixation, the nitrogen which is present in the atmosphere, it undergoes reduction and it leads to the formation of ammonia. The enzyme which carries out this process is the nitrogenase enzyme, and this nitrogenase enzyme, it is present in case of prokaryotes, and it will help in the fixation of this nitrogen. Now, this enzyme is capable of reducing nitrogen, and it is present exclusively in case of prokaryotes. So, in prokaryotes, this enzyme is exclusively present. Such microorganisms, they are also known as nitrogen fixers. So the reaction which is taking place in case of prokaryotes for the fixation of nitrogen is as follows. That is N2, that is the nitrogen molecule which is present in the atmosphere, by the help of enzyme nitrogenase, it gets converted into ammonia. This is how fixation of the nitrogen will take place. The nitrogen fixing microorganisms, they may be free living or they may be symbiotic, means they may be free living in the soil, or they may be associated with the roots of the plant. They are known as symbiotic association. So these nitrogen fixing bacteria or microorganisms are of two types: free living and symbiotic. Some examples of free living nitrogen fixing microorganisms are as follows. Now, in case of nitrogen fixing microorganisms in aerobic class, the two examples are Azotobacter and Biogernicia, whereas the anaerobic nitrogen fixing microorganisms, that is in absence of oxygen, the example is Rhodospirillum. It is an anaerobic microorganism which can fix the nitrogen. Bacillus and cyanobacteria such as Anabina and Nostoc are another examples of free living nitrogen fixers. So nitrogen fixers, it may be fixing nitrogen in presence of oxygen, or they may be capable of fixing nitrogen in absence of oxygen, and that's why there are two different types of nitrogen fixers: aerobic and anaerobic. Now, as we have discussed that nitrogen fixing microorganisms, they could be found free living in the soil, or they would be symbiotic also. They can be symbiotic also. So here there is another topic that is known as symbiotic nitrogen fixation and nodule formation. So if you consider symbiotic nitrogen fixation, we have another example of a microorganism which is known as rhizobium bacteria, and this is one of the well-known example in case of symbiotic nitrogen fixation. Now, rhizobium bacteria, it forms root nodules in case of leguminous plants such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans. So basically these are all leguminous plants, and this rhizobium bacteria, it can form root nodules in the leguminous plants. So this is one mechanism of fixing atmospheric nitrogen, that is nodule formation will help in fixing atmospheric nitrogen in the form of ammonia, and plants can take that nitrogen easily. Now there is another example, that is Frankia. it produces nitrogen fixing root nodules on the roots of non-leguminous plants such as alnus. So these are the two examples, that is rhizobium, it forms root nodules in leguminous plants, and frankia, it produces root nodules in non-leguminous plants. So this is how the nitrogen fixation can take place in leguminous plants as well as non-leguminous plants by different kind of bacteria. One is rhizobium and the another one is frankia. Both rhizobium and frankia, they are free living and they are capable of fixing the atmospheric nitrogen. Now when we cut the nodules, we can see a pink color or red color in the center. So this pink color or red color is because of leg hemoglobin. Also it is known as leguminous hemoglobin. Now we will go with the importance of this leguminous hemoglobin when we discuss this nodule formation in detail. So these nodules will be containing leg hemoglobin or leguminous hemoglobin. So this is very important with respect to the examination point of view. Now, nodule formation, it involves various or sequence of interactions between rhizobium and the roots of the host plant. So basically this nodule formation is because of the interaction between the rhizobium bacteria and the roots of leguminous plants. Now what happens in case of nodule formation is the rhizobia, it starts multiplying and it starts to form colony around the roots or surrounding the root hairs. So to the surrounding of the root hairs, you can see that large number of rhizobia will be produced. Now they will attach to the root hair cells and they will lead to the curling of the root hairs. So as soon as they attach to the root hair cells, they will lead to the curling formation of the root hair. So if you see the diagram which has been shown here, you can see that there is presence of root hair, and around root hair there are large number of bacteria which are present. Now what this bacteria will do, these bacteria will divide, they will form colony. As a result, what will happen, they will lead to the formation of a curl or a hook like character in root hair. So this is the hook formation or curling of the root hair. Now as soon as the root hair curl or hook like character is formed in the root hair, then the bacteria will start producing an infection thread. So an infection thread is produced in the root, and due to this infection thread, the bacteria, it goes into the cortex of the root. So here you can see in the third part of the diagram, an infection thread is being formed where the bacteria through the root hair, it goes to the inner cortex and the pericycle cells of the root. So this is basically due to the infection. An infection thread is being produced. So now the infection has been created into the root cells, that is the inner cortex or pericycle cells. Now bacteria, when they reach the cortex, they start the nodule formation in the cortex of the root. The bacteria, they get released from the thread into the cortex cells, in which they will differentiate, and they differentiate into special kind of cells known as nitrogen fixing cells. Now these bacteria causes the cortical cells and pericycle cells to divide further, and due to large number of division of cortical cells and pericycle cells, a nodule is being formed. So basically this is important, that bacteria, it leads to division of cortical cells and pericycle cells, which leads to the nodule formation. And when this bacteria reaches to the cortex, it gets differentiated into a special kind of nitrogen fixing cell. Now this nodule, it also develops connection, a vascular connection with the host plant, so that it can get the nutrition also. Now the nodule, it contains a special enzyme which is known as nitrogenase. Now as students, we have gone earlier that the enzyme nitrogenase, it is capable of converting the nitrogen molecule into ammonia. So this nodule, it basically contains the enzyme nitrogenase. This enzyme nitrogenase is a molybdenum iron protein, and it helps in the conversion of atmospheric nitrogen into ammonia. So conversion of atmospheric nitrogen into ammonia, it takes place inside the nodule by the help of enzyme nitrogenase. Now this nodule contains specific protein which is known as leg hemoglobin. So as we have discussed that if you cut the nodule, you can see a pink or red colored substance inside it, which is known as leg hemoglobin. It is a special kind of protein which acts as an oxygen scavenger. Means this leg hemoglobin will try to consume the oxygen. As a result, it will provide anaerobic condition to the bacteria, because nitrogenase enzyme, it cannot act in presence of oxygen. It is very sensitive to oxygen, so it needs anaerobic condition. So to create anaerobic condition, leg hemoglobin is present in the nodule. So this leg hemoglobin will act as an oxygen scavenger. It will absorb the oxygen amount, it will absorb the oxygen, and it will provide an aerobic condition for the nitrogenase enzyme to convert nitrogen into ammonia. So this is the reaction which takes place in the conversion of nitrogen into ammonia. We can see that for the two molecules of ammonia to be produced, 16 ATPs are required. So for one molecule of ammonia to be produced, 8 ATP molecules are necessary. So here there is an stepwise conversion of atmospheric nitrogen into ammonia by nitrogenase enzyme, where we can see that at the starting point there is the nitrogenase enzyme which undergoes combination, or which combines with the substrate, that is nitrogen gas, that is N2. So this undergoes reduction three times. There are three sequential reduction which takes place. As a result, what will happen? The triple bond between the nitrogen will break sequentially, and one nitrogen atom will combine with three hydrogen atoms due to three different reduction reactions, and at the last, two molecules of ammonia are released, and the enzyme, it is also released, as it can be used once again in the same way. So this is how there is a stepwise conversion of nitrogen into ammonia. Now what happens to the ammonia? That is, what is the fate of ammonia? Now at physiological pH, ammonia, it gets converted into ammonium ions by protonation. So ammonia, it combines with a proton to give ammonium ions, and this ammonium ions, it is used to synthesize the amino acids in the plants. So basically there are two ways in which this process can take place. The first one is known as reductive amination. Here in this process, ammonia, it reacts with alpha ketoglutaric acid. So this ammonium ion, it is reacting with alpha ketoglutaric acid in presence of NADPH, in presence of enzyme glutamate dehydrogenase, leading to the formation of glutamic acid, and this glutamic acid can be used further by the plants. The same way, there is another reaction which happens here, is transamination. In transamination, there is transfer of amino group from one amino acid to the keto group of the keto acid. So here the amino group, it gets transferred from one amino acid to the keto group of the keto acid, which has been shown in the reaction below. So there is an amino donor from which the amino group will go to the amino acceptor part, where you can see that the amino group, once it is donated, it leads to the formation of ketone group, and the amino acceptor, after accepting the amino group, it leads to the formation of the ammonium ions. So glutamic acid is the main amino acid which is formed due to the transfer of amino group that takes place, and the other amino acids are formed through the process of transamination. The enzyme transaminase will help in conversion of this particular reaction, or it helps in the process of transamination. Now there are two types of amides, asparagine and glutamine, which are also formed from aspartic acid and glutamic acid by addition of one more amino group to the amino acid. So if you add one amino group to the amino acid, it leads to the formation of two amides, that is asparagine and glutamine. So friends, with this we complete this chapter. In the next class we'll start with the another chapter. Thank you very much.